Hello, biologists. Welcome to another edition of BioCasts. Now that we're in Chapter 18, let's start talking about viruses. A quick reminder, all the slides you're going to see in this podcast are available as a notes handout on the AP Biology page of mrtraphagen.com. So, if you have a printer and haven't printed them yet, please pause the podcast and print these before going forward. Okay, so let's start talking about what viruses are. Viruses are small, really, really small. In this diagram, you see a eukaryotic cell, okay, an animal cell. You can see it's got a nucleus, it's got a mitochondrion, okay. In the foreground, that little flagellated thing is a bacterial cell, much smaller than the eukaryotic cell. Above the bacterium, that little insect-looking thing is a virus, okay. They are much, much smaller than bacteria. They are capable of reproducing as long as they're in a host cell, and they are constantly evolving, which is why every year we have to get a flu shot, because medical scientists try to guess what the influenza virus is going to look like the upcoming winter. Every year they evolve, every year there's a new flu virus, every year there's a new flu shot. That brings us to what viruses are not. They're not cells. They don't have organelles, they're not capable of doing metabolism on their own, they're not capable of reproducing on their own. In order for viruses to reproduce themselves, they have to hijack the cellular machinery, the organelles of a living cell. Because they can't do these things on their own, we say viruses are not alive. I cannot stress that strongly enough. We draw the line between bacteria, which are really small, simple cells, those are alive, and viruses, which are not alive. Okay, let's do a quick history lesson on how viruses were discovered. In the late 19th century, Adolf Meyer, a German scientist, was studying this disease affecting tobacco plants. You can see this disease on the leaves in diagram B on this slide. Adolf Meyer took the sap from an infected plant and rubbed it onto the leaves of a healthy plant, and he made those healthy plants sick. We now know that this problem was caused by something called the tobacco mosaic virus, but at the time they didn't know what this was. So, skipping some of the details of who studied what, let's just ask the two important questions. We have something making these tobacco plants sick. Why not bacteria? Why not toxin? So, their first guess was that this was a bacterium. Okay, they did microscopy on the sap, found no bacterial cells. They then filtered that sap through a microscopic filter that was small enough to filter out bacteria. Then they took the filtered sap, put it on new healthy plants, and they still got infected. Okay, so we have some pretty decent evidence that it's not bacteria, but could it just be some small molecule toxin? To test this, they took sap from an infected plant, which has this problem in it. Maybe it's a toxin. They take that infected sap, put it on healthy plants, they get sick. They wait a while, take those now infected plants, rub their sap onto yet another plant, that plant gets sick. And they did this over and over and over again. If it were a toxin, we would expect it to get diluted each time they passed it through a new plant. But what happened was, every time they passed the sap to a new plant, the new plant got sick. So somehow this thing that was making the plant sick was reproducing itself. So we know it's reproducing, but we know it's smaller than a bacterium. This is something new. Okay. So I've been telling you, and it's an oversimplification, that you can think of a virus like an M&M. The thin, colorful candy shell is the protein coat, which now we're going to say is called the capsid. The protein coat is called the capsid. Inside that protein coat, you have some nucleic acid. Maybe it's DNA, maybe it's RNA, but it has to be a nucleic acid. You can see that these viruses take different shapes. From left to right, we have an RNA virus, the tobacco mosaic virus, that's cylindrical in shape. Okay, the adenovirus, second from the left, has a really cool geometric shape to its capsid. Okay, you can see the capsid is made up of these triangular faces. Okay, the influenza virus, the one we know of as causing the flu, okay, has a spherical capsid. And then all the way on the right-hand side, we have a bacteriophage, or just phage, okay, that has this interesting insect-looking shape. Okay, let's pause now for a thought question. After I ask this, I want you to pause your podcast, write down your thoughts. This goes back to our assignment at the beginning of the year. What is the definition of alive? So if viruses are not alive, how can we say they evolve? That's something we typically only talk about living things doing. And if viruses are alive, where do we draw the line? Are computer viruses alive? Are chain emails alive? Where do we draw this line between alive and not alive? Pause the podcast, and I'll join you on slide 9. Okay, welcome to slide 9. Let's talk about how viruses reproduce themselves, or rather, get cells to reproduce their viruses. In order for a virus to reproduce, it has to invade a living cell. It then has to transcribe and translate its nucleic acid. Notice I put bracket NA. You might use RNA to make a new virus, you might use DNA, but you're going to transcribe that DNA into RNA, and you're going to then have to translate that RNA into protein because you're going to need to make more protein capsid to contain your new viruses. To do that, you're going to have to hijack the cell's ribosomes. Once you have your new capsids assembled, you have your replicated nucleic acid in the new capsids, you've made a whole bunch more virus. Now you're going to have to lyse the cell, and those viruses spread out to find new cells to repeat this cycle. Now, this is all viruses do. No other metabolic processes. All they do is reproduce their genetic material and move on. This is why we say they're not alive. Okay, so the viral, I'm doing air quotes as I record this, life cycle. There are two basic strategies viruses use. There's something called the lytic cycle and something called the lysogenic cycle. Now, reminder foxes here because you need to know the difference between these two and you need to know the names. The lytic cycle, the virus invades the cell, reproduces as fast as possible, and then immediately splits that cell open, looking for new cells. The lysogenic cycle, though, is a slower process where the virus replicates its nucleic acid in the host cell, but does not finish its assembly and split the cell open. The lytic cycle are what we call virulent viruses. These are the viruses you'll notice making you sick. They cause massive cell death because every cell that gets infected is split open by this rapid reproduction of lots of new viruses. The lysogenic cycle, on the other hand, is more like a time bomb. The HIV virus does this. This is one of the reasons people infected with HIV appear to show no symptoms for many years before all of a sudden they start to get sick. Here we see the lysogenic cycle with a phage virus. Okay, remember, phage are viruses that attack bacteria, and phage literally means to eat. So these are viruses that only target bacteria. So you see the phage starting on the upper left side. The phage virus inserts its DNA into the bacterium. The bacteria then replicates that phage DNA multiple times. It gets incorporated into the cell, and it keeps replicating it. And as it goes through that lysogenic cycle, every time that bacteria reproduces, you get an offspring that includes that phage DNA. Now, on the left side, you see the lytic cycle. This is the immediate, explosive, kill the cell, make as many viruses as you can, and move on. Okay, some fun facts about viruses. You knew this already. They attack very specific cells. If you get a cold, that cold doesn't infect your muscles. It infects specifically the epithelial or skin tissues of your respiratory tract. You get a cold in your head or in your chest. You don't get a cold in your muscles or your feet. The polio virus, not as big a problem anymore now that we have a vaccine, used to be a very serious problem, specifically attacks the nerve cells. Since nerve cells aren't very good at reproducing or repairing themselves, the poliovirus tended to cause lifelong symptoms like paralysis. Once you had those nerves damaged, they weren't coming back. And then HIV, or the human immunodeficiency virus, attacks one specific kind of white blood cell called the helper T lymphocyte. So all three of these viruses attack humans, but they attack different types of human cells. More fun facts about viruses. Bacteria can fight off their viruses, phage, without using immune systems. Remember, bacteria, they're so simple, these little single-celled critters. They don't even have membrane-bound organelles. What they do have are special enzymes called restriction enzymes that cut specific DNA sequences. So, for example, every time the restriction enzyme in E. coli bacteria sees a GGCC sequence of DNA, it will cut that DNA backbone. Since bacteria are small, E. coli don't have any GGCC sequences. So anything that has GGCC must be an invading virus. Restriction enzymes are used by bacteria to fight off their viruses. We have now stolen these restriction enzymes to do medical research. You'll hear more about that in podcast number 4. Okay, we've reached the end of podcast number 3. Before you put this away, make sure you write down any notes, questions, concerns while they're still fresh in your mind. You want to use your notes from this podcast and your notes from the Chapter 18 reading notes to make sense of this viral stuff. Okay, at this point, I also want to note that BioCast number 4 is also posted online. Make sure you watch both of these before class on Monday. Good luck, AP biologists.